Okay, so welcome back to this next video in which we are discussing depression and the antidepressant drugs. Okay, so we're currently in the process of discussing the monoamine hypothesis of depression, which is the hypothesis that depression is caused by too low levels of monoamines within the brain. Specifically, serotonin and noradrenaline are believed to be involved, particularly serotonin. Serotonin is the famous one associated with depression. Okay, now, the big flaw with the monoamine hypothesis, well actually first let me tell you what's really in the favour of the monoamine hypothesis. The big thing in favour of the monoamine hypothesis is that all the antidepressant drugs work by increasing the amount of serotonin or noradrenaline or both within the brain. Okay, so that seems really powerfully in favour of the monoamine hypothesis. The flaw is that all you need to do is take one tablet in order to raise the levels of serotonin and noradrenaline within your brain. Okay, but how long does it actually take for these drugs to have an antidepressant effect? Well, you don't get the antidepressant effect as soon as you take one tablet. They take weeks of taking the tablets to actually produce an antidepressant effect. That doesn't make sense. If depression is caused by too low monoamines, then surely by replacing the monoamines, the depression should go. You should get alleviation as soon as you take one tablet, basically. Okay, and that's not the case. So that's a flaw in the monoamine hypothesis. The general view now, I think, is that the antidepressants do raise the levels of serotonin and noradrenaline within the brain, and that this raised level of serotonin and noradrenaline within the brain then causes changes in the brain, which then require a few weeks to actually occur, and that those changes in the brain are then what bring about the antidepressant effect. Okay, so potentially they might change the overactivity in the anterior cingulate cortex. So let's say we believe that overactivity in the anterior cingulate cortex is responsible for depression, then potentially by raising the monoamines, by raising serotonin and noradrenaline, you might reduce that overactivity in the anterior cingulate cortex. But it might require weeks and weeks of having raised serotonin and noradrenaline to do that. Okay, so that I think is the general view now. Okay, right. However, we will continue on with the monoamine hypothesis. Okay, we're going to continue on by discussing further the structure of serotonergic neurons and noradrenergic neurons. We're going to look at how the neurotransmitters are synthesized, how they're packaged into storage vesicles, okay, and then how they're reuptaken after release, okay, because understanding of that is going to be really important in understanding the way the antidepressants work, and also in understanding a few of the drugs that led to the formation of the monoamine hypothesis in the first place, because there were two original drugs that really solidified the monoamine hypothesis in place. Okay, and I want to discuss how those work, and that requires a better understanding of the serotonergic and noradrenergic neurons. Okay, right. So we will start off with the serotonergic neurons. Okay, so the neurons which release serotonin. Okay, so I'm not going to draw the whole neuron. All we're going to actually need is the axon terminal. Now I'm going to draw it very, very large because I want to draw a lot inside this axon terminal. Okay, right. Well, actually I'm not going to be able to draw the synthesis inside this axon terminal. We're just not going to have space. It's not big enough for that. But there we go. Here's our picture of an axon terminal. Okay, so we're going to start off with how do you synthesize serotonin within these axon terminals. Okay, well it's synthesized from the amino acid tryptophan. Okay, so let's start by discussing the structure of the amino acid tryptophan, and then we'll see what modifications are going to occur to the amino acid tryptophan to turn it into serotonin. And remember, the other name for serotonin is 5-hydroxytryptamine. Okay, so we're starting to see links potentially with tryptophan already. Okay, right, as soon as we use that name. Right, so let's start then with the structure of tryptophan. So, tryptophan then, Tryptophan has within its structure an indole ring. Okay, so I firstly want to show you the structure of an indole ring before I show you the structure of tryptophan. Now, in order to discuss what an indole ring is, we firstly need to discuss what a pyrrole ring is. So we'll start off with a pyrrole ring. Okay, now I'm going to show this in skeletal formula. So remember, in skeletal formula you do not show carbon atoms. They are implicitly shown by corners. You also do not show hydrogen atoms attached to carbon atoms. Okay, where you have missing bonds coming off carbon atoms, those are implicitly 2 hydrogen atoms. Okay, right. So a pyrrole ring is a 5-membered ring. Okay, 4 of the members are carbons, so they're just being shown as corners, and then the 5th member is a nitrogen atom, right up there. Okay, so that's a nitrogen. It is going to have a hydrogen coming off. This is a little bit difficult to show, it's a little cramped, but there is a hydrogen coming off that nitrogen there. Okay, now there are also 2 double bonds. There's a double bond here, and there's also a double bond here. Okay, now that leaves these 4 carbons here, this one, this one, this one, and this one, with another bond that is free. Okay, each of those carbons will have a bond to a hydrogen atom, but we don't need to show that because we're drawing a skeletal formula. Okay, so this ring structure now, this is what's known as a pyrrole ring. Okay, we're now going to upgrade slightly. We're going to go over to an indole ring. Now, an indole ring is going to contain a pyrrole ring, which is why I've started off by showing you a pyrrole ring. So let's just redraw out our pyrrole ring again. So here is our 5-membered ring structure, okay, where 4 of the members are carbons, and then 1 of the members is a nitrogen. Okay, like so, and then we need a hydrogen coming off that nitrogen. Okay, right, and now what we're going to do is attach this to a benzene ring. So we're going to have a 6-membered ring attached on like this. Okay, and this is going to be a benzene ring, so we're going to have alternating double and single bonds in this additional ring here. Okay, so we've got a double bond, single bond, double bond, single bond, double bond, single bond. And again, each of these 4 carbons here will have a hydrogen coming off it. Okay, so that now is the structure of an indole ring. Okay, and this is the structure that we are going to put in tryptophan. Okay, so let's now show tryptophan. Okay, I've drawn that indole ring in a really inconvenient place. It should have been here. Is that space now big enough to try and squeeze tryptophan in? I'm not going to try. I'm going to put it sort of here. No, here. Okay, so tryptophan then, it's an amino acid, so I'll start by drawing the core amino acid structure that all amino acids have. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, and here's the carboxylic acid group down here. Okay, that's the core amino acid structure that all amino acids have. Okay, then the R group of a tryptophan amino acid contains a methylene group. Okay, and then off the side of it, you then have an indole ring. Now, how are we going to attach an indole ring on here? Well, we're going to use this carbon here, okay, of the pyrrole ring within the indole ring. Okay, so the whole thing, by the way, the whole thing is called an indole ring. I know it's really 2 rings, okay, but the whole thing is called the indole ring. Okay, we're going to use this carbon here. We're going to take the hydrogen off that carbon, and then we're going to attach it to this carbon here. Okay, right, so here is the pyrrole ring, like so. The nitrogen will have a hydrogen coming off there, and we've got double bond here, and then we've got the benzene ring coming off here. Double bond there, double bond there. Okay, and this structure here, this is now tryptophan. Okay, right. The amino acid tryptophan, so I'll underline that in orange here. And it's smudged terribly again. Right, okay. So, this is the amino acid tryptophan. This is the starting point for the synthesis of serotonin. Okay, right. So, the first alteration we are going to make is we're going to add an alcohol group onto the 5th position. So we are firstly going to convert tryptophan into something known as 5-hydroxytryptophan. Okay, and I'm going to show you exactly where we are going to add on this alcohol group to turn this into 5-hydroxytryptophan. Okay, so we've quite close now to serotonin, because remember, serotonin was just 5-hydroxytryptamine. Okay, so we're only one step away once we've got to 5-hydroxytryptophan from 5-HT itself. Okay, and by the way, 5-hydroxytryptophan is often abbreviated to 5-HTP. So if you see 5-HTP, that means 5-hydroxytryptophan rather than 5-hydroxytryptamine. Okay, right. So all we're going to do is stick an alcohol group onto the 5th position of the tryptophan molecule. So what we need to know is, where is the 5th position? So I'm going to tell you something about the naming for the members within the structure of tryptophan. So basically we'll start here, okay? This is the alpha carbon, okay? Because it's the first carbon that comes off the carboxylic acid group. Okay, following that, this second carbon here, this is known as the beta carbon. Okay, so that's the naming of these two carbons here. Okay, you'd call this the carboxylic acid carbon, and this the amino group. Okay, now let's talk about the members of the indole ring. So in the naming of the members of the indole ring, you start with the nitrogen, and you call that number 1. Okay, so you use a completely different naming system to alpha and beta here. Okay, you start with the nitrogen, you call that number 1, and then you go round. So this one is number 2, this carbon that's smudged horribly here. Okay, this carbon here is number 3. Okay, now you would think this would be number 4, but someone decided that when a carbon is in between two rings like that it shouldn't be given its own number. Okay, so instead of being called 4, it's called 3A. So you take the same number as was on the previous one and you just put an A in front of it. So that's 3A. Okay, then this one's not in the middle of two rings, so this is 4. Okay, then this is 5, this is 6, this is 7, and then again this one is in the middle of 2, so it's not called number 8, but it's called 7A instead, and I'm not totally sure what the logic behind that is. If anyone knows, please do leave a comment. Okay, right. Now, that's answered the question though. That tells us exactly where number 5 is. That is where we are going to stick an alcohol group on, up there. So that carbon has a hydrogen off it, okay? Not shown, but it does have a hydrogen coming off it. So we are going to remove that hydrogen that comes off that carbon, and we're going to replace it with an alcohol group. So let's show this having happened. So here is the amino group, so I'll draw the full thing out again. Okay, here is the alpha carbon with a hydrogen coming off it. Okay, here is the carboxylic acid group here. Okay, then the R group now of 5-hydroxytryptophan. It has this methylene group still, and now let's draw the pyrrole ring again. So here are the 4 carbons of the pyrrole ring. Here is the nitrogen atom. Okay, it will have a hydrogen coming off. Here's a double bond, here's a double bond. Okay, and then we'll have the benzene ring up here. Okay, we'll have a double bond there, and now we've got an alcohol group coming off this 5th carbon here. Okay, and that then is the structure of 5-hydroxytryptophan, or 5-HTP. Okay, right. So, what catalyzes the addition of this alcohol group onto the 5th carbon of the tryptophan amino acid? Well, it is an enzyme known as Tryptophan Hydroxylase. Okay, so it's nice and sensibly named. You could have guessed that. Okay, Tryptophan Hydroxylase. Okay, which is just the standard name for an enzyme which adds hydroxyl groups onto things. So tryptophan hydroxylase is the enzyme which will catalyze this conversion. Okay, and that's occurring in the cytoplasm. So tryptophan in the cytoplasm is being converted into 5-hydroxytryptophan. Okay, now another conversion is going to occur now. Okay, we are going to convert 5-hydroxytryptophan into 5-hydroxytryptamine. We're going to turn it into serotonin, basically. And the way we're going to do that is by removing the carboxylic acid group here. Okay, so this carboxylic acid group, it's going to be removed, okay, and replaced with a hydrogen atom there instead. Okay, so I'll draw the end product that you get here. So here's the amino group. Okay, here's the alpha carbon with a hydrogen coming off it, but now it's got a new hydrogen coming off it. Okay, it's got 2 hydrogens coming off it now. Okay, then the R group is going to consist of a methylene group. Okay, and now we have our indole ring. Okay, so we'll start with the pyrrole ring here. So here's the nitrogen. Okay, here's the hydrogen coming off that nitrogen. Double bond there, double bond there. And then we'll have the benzene ring. And then we're going to have the alcohol group coming off the 5th position of the benzene ring. Okay, up here. Okay, and this structure now, this is 5-hydroxytryptamine. Okay, or just 5-HT, or of course you can call it by the more popular name, which is just serotonin. Okay, right, so 5-hydroxytryptophan is going to be converted into 5-hydroxytryptamine then. So what is the name of the enzyme which catalyzes this conversion? Okay, well the name of the enzyme that catalyzes this conversion is the L-aromatic amino acid decarboxylase. Okay, so it's a decarboxylase because it removes carboxylic acids. It removes carboxylic acid groups from things, so decarboxylase. Okay, and we're going to see this enzyme again. It's going to function in the synthesis of noradrenaline as well, and also dopamine. Okay, because dopamine is the precursor to noradrenaline, so it will be in the same pathway. Okay, so for short, L-aromatic amino acid decarboxylase is abbreviated to L for L, A for aromatic, A for amino acid, okay, and then D for decarboxylase, so the LAD enzyme. Okay, so again, this enzyme's in the cytoplasm and will convert 5-hydroxytryptophan into 5-hydroxytryptamine in the cytoplasm. Okay, so I just want to make sure you understand what an L-aromatic amino acid is. So aromatic's easy. That just means that the R group contains an aromatic structure. An indole ring is an aromatic structure, so that's fine. Okay, L-amino acid then. What's an L-amino acid? Okay, most amino acids have 2 optical isomers. Okay, so I'm going to go over the page to explain this. So, if I draw the generic structure of amino acids, you have an amino group here. Okay, and this is of the proteonergic amino acids, so the amino acids used in proteins. You have the alpha carbon here with a hydrogen coming off it, and the carboxylic acid group here. Okay, and then also coming off this carbon, you're going to have the R group. Now, if that R group is not a hydrogen, okay, which is the only case that you can have where you don't have 4 different groups coming off here. Okay, providing this R group is not the same as one of these, and it will never ever just be equal to an amino group, and it won't ever equal the carboxylic acid group. There are no proteonergic amino acids where the R group is the same as this group or the same as this group. The only possible way that this can happen is that the R group can be a hydrogen. There isn't a proteonergic amino acid where the R group is just a hydrogen, that's glycine. But apart from in the case of glycine, okay, all the other proteonergic amino acids have 4 different groups coming off this carbon here, okay, which means that that carbon is very special, okay. It's what's known as chiral, sorry about this, okay, it's what's known as a chiral carbon, okay, or a chiral center, okay, and this means that it's going to give rise to optical isomers. So there are going to be 2 different optical isomers of all amino acids, all proteonergic amino acids apart from glycine. Okay, and I want to explain why this is. Okay, so to explain why this is, you have to understand that this picture is a 2-dimensional representation. Okay, if we start drawing 3-dimensional representations, hopefully you'll see why 2 different forms of this molecule can arise. Okay, so let me put that alpha carbon right at the center here. Okay, and I'm also going to put the hydrogen that comes off the alpha carbon, this hydrogen here, pointing upwards. So imagine that we put the carbon, okay, in the plane of the piece of paper, and we also have the hydrogen in the plane of the piece of paper as well. Okay, then let's imagine that we put the amino group in the plane of the piece of paper as well. We can do that. We can choose to make sure that these three atoms here, the hydrogen, the carbon and the nitrogen here, are all in the plane of the piece of paper. However, the final two groups that are coming off here, the R group and the carboxylic acid group, those cannot stay in the plane of the piece of paper. One of them is going to come out of the page towards us, off this carbon, at this sort of angle that I'm demonstrating to you now. The other is going to go into the page away from us at this sort of angle that I'm demonstrating now. Okay, now the way that I show bonds coming out of the page towards us is by making them thicker towards the edge, like so. Okay, and it's not just my way of doing it, many people use this way. Okay, so that's the way of showing a bond that's coming out of the page towards us. The way of showing a bond that's going into the page away from us is like so, with a dashed line. Okay, so this is now a three-dimensional structure. Now then the question becomes, do I have the carboxylic acid group going away into the page away from me, and the R group coming out of the page towards us? Or do I have the carboxylic acid group here, okay, and the R group going into the page away from us? Okay, so those two options, and I'll draw the other one here. Okay, so let me put the other one here. Okay, like so. So again, I'll have the amino group, the carbon, and the hydrogen in the plane of the piece of paper. Okay, but this time, coming out of the page towards us, okay, is going to be the R group, okay, and going into the page away from us is going to be the carboxylic acid group, okay. These two molecules are fundamentally different. You cannot turn this one into this one without performing a chemical reaction. You would have to break this bond, swap the groups around, and then you'd have this. Okay, without performing that chemical reaction, you cannot turn one into the other. Okay, there's no way of just changing the way that you look at this to make it look like this. They are fundamentally different molecules. Okay, and they are known as optical isomers of one another. Okay, because they are incredibly similar. Okay, the reason they're called optical isomers is because in the case of these, they're actually very, very beautiful optical isomers of one another. Okay, they're actually special types of optical isomers known as enantiomers. Okay, now not all optical isomers are this easy. Okay, other optical isomer situations are much more complicated than this. But in the case of enantiomers, the two different optical isomers are actually mirror images of one another, and that's the origin of calling this optical isomerism, because with a mirror you can turn one into the other. Okay, but not just by rotating it, you can't turn one into the other. Okay, so enantiomers are when the two optical isomers are mirror images of one another, and that's not always the case in optical isomerism. Okay, so they're mirror images of one another. Okay, so basically you might be wondering, well, how is this the mirror image of this? If you imagine this one looking in the mirror, okay, it would actually see this one rotated around. Imagine rotating this one around 180 degrees. Okay, the amino group would now be pointing away in this direction, but it would still be in the plane of the piece of paper. The R group would now be going away from us into the page down here, and the carboxylic acid group rotated around 180 degrees would be coming out of the page towards us down here, and that's the mirror image of this here. Okay, so this one here is the mirror image of this one's the mirror image of this one. Okay, so they're very, very special optical isomers. They are enantiomers. Okay, so all amino acids have these two forms. Okay, now there is a naming for the two different forms. Okay, and this one here is called the L enantiomer, or the L amino acid. So we can call this the L enantiomer. Okay, and this one is called the D enantiomer. Okay, now L and D stand for very long words. Okay, so L stands for larvo-rotatory enantiomer. Okay, so that's what L stands for. And D stands for dextro-rotatory. Okay, right, so how do you remember which amino acid is the L one and which is the D one? Well, there is a useful trick. Okay, if you imagine you are the little hydrogen, okay, sitting up there and looking down at these three groups, Okay, try and read the word corn. Okay, now how are you going to do this? Well, you're going to read the CO from the carboxylic acid group. You're going to read the R from the R group. Okay, and you're going to get the N from the amino group. Okay, now if you were to read it, if you were to read corn in the case of the L enantiomer, your head would go from here, the CO, to the R group, to the N. Now it would be moving in a clockwise direction. Okay, so if you're sitting at the where the hydrogen is, and you're trying to read the word corn, and your head is going in the clockwise direction, then you are sitting on top of the L enantiomer. Okay, whereas if you try and read corn on the dextrorotatory enantiomer, then you go from the CO here, okay, to the R, to the N. Okay, and this time, what direction is your head going to go in? It's going to go from the carboxylic acid group to the R group, to the amino group, and then well, that's it, finished. Okay, it's going in the anti-clockwise direction. So the dextrorotatory enantiomer is the one where your head has to move anti-clockwise, okay, to read the word corn, basically. Okay, right, so that's a simple way to remember whether it's an L enantiomer or a D enantiomer. Okay, now, all the amino acids, pretty much, that are used in cells are the L type. Okay, we don't really use the D enantiomers at all. Okay, so that makes it simple. Okay, but that's the origin of the naming of that enzyme. It's just referring to the fact that the enzyme will only catalyze this providing that the 5-hydroxytryptophan is in the L form, but it will be, basically. Okay, so that's all that that's referring to. So that was a very long explanation for that simple naming curiosity. Okay, I should have just passed it off as a naming curiosity. Right, okay, so we now have synthesized 5-hydroxytryptamine. Okay, what I'm now going to describe to you is how we're going to package the 5-hydroxytryptamine into vesicles. So now we'll go back to this picture here. So in the axon terminal, okay, you have these little membrane-bound spaces known as vesicles, okay, and this is going to be filled up with neurotransmitter, and for that reason it's known as a synaptic vesicle. Okay, so we need to get the 5-hydroxytryptamine, which is currently in the cytoplasm, into the vesicle. So how is this done? Well, it's done using a transporter, okay, which I'll put here. And this transporter, which I'm going to colour in purple, is known as the vesicular monoamine transporter, or for short it's abbreviated to VMAT. Okay, so this is the vesicular monoamine transporter. Okay, so V for vesicular, M for mono, whoops, I've spelt that wrong, A for amine, and then T for transporter. Okay, right. So there is not just one type of vesicular monoamine transporter, there are actually two different types. Okay, there is a VMAT1, okay, and then there is a VMAT2. The main form that is found within the brain is the VMAT2 rather than the VMAT1. Okay, and I just would like to describe something about these transporters, which I'll do over the other page. So, these transporters, VMAT1 and VMAT2, are actually in a bigger family of transporters known as the solute carrier transporters. Okay, so there is a massive great family of transporters in biology known as the solute carrier transporters. We are going to come across more solute carrier transporters later on. Now, there are a huge number of different families of solute carrier transporters. VMAT1 and VMAT2 are in the 18th family. Okay, so they are in SLC18. So SL is short for solute. Okay. C is for carrier, and the 18 is then denoting that we are talking about the 18th family of solute carrier transporters. Okay, so in this 18th family there are three main members. Okay, so let me outline these three main members. They are SLC18A1, okay, SLC18A2, okay, and then finally SLC18A3, okay. Right, now SLC18A1 is the VMAT1, okay, and it's a separate gene to the gene for VMAT2, which is SLC18A2. So those are the fancy names for VMAT1 and VMAT2, SLC18A1 and SLC18A2. Okay, the SLC18A3 does a very similar thing to SLC18A1 and A2, okay, but it does it for a different neurotransmitter. It does it for acetylcholine, so it's known as the VACHT, okay, the vesicular, that's the V, and then the ACH is for acetylcholine, and then the T is for transporter. So this is a transporter which transports acetylcholine into synaptic vesicles. Okay, right, so these three different members of this SLC18 family, they all have the same basic structure, which I'm going to outline for you now. Okay, so they are proteins with 12 membrane-spanning alpha helices. So they'll have the amino group here. Okay, so they are individual proteins. That's the first thing to say, actually, that each one of these is an individual polypeptide. It's not a complex of polypeptides. Okay, it's an individual polypeptide. So here's the first membrane-spanning alpha helix, the second, the third, the fourth, the fifth, the sixth, the seventh, the eighth, the ninth, the 10th, the 11th, the 12th. Okay, so let me just explain the picture that I've just drawn for you. Okay, so these two lines here, these represent the lipid bilayer that makes up the membrane of the synaptic vesicle. So this synaptic vesicle that I drew here, this is actually a phospholipid bilayer, so it's made up of two layers of phospholipids. Okay, so this is the lipid bilayer. And then implanted in this lipid bilayer, you have one of these transporters. Okay, now if we're talking about the vesicle within a neuron in the CNS, it's likely to be the SLC18A2 or the VMAT2. Okay, VMAT1 does the same thing, but it does it generally in neuroendocrine cells. So for instance, the cells of the adrenal medulla that make adrenaline and store it in vesicles as well. Okay, so it will perform the role in those cells rather than the central nervous system neurons. Okay, now this side of the membrane is the cytoplasmic side. So the side with the amino and carboxylic acid groups on is the cytoplasmic side, and this is the vesicular side. So this is the vesicular lumen. Okay, so that's what these proteins look like basically. This is what VMAT2 actually looks like. It spans the membrane of the vesicle several times. It's an integral membrane protein in the vesicle membrane. Okay, and when it spans the membrane, these points where it spans the membrane are alpha helical structures. Okay, so they look like springs basically. They look like this. Okay, even though I haven't shown that in this case, I've just drawn them as lines. That they are in fact alpha helices. Okay, they're known as membrane spanning alpha helices. Okay, right. So you have this VMAT2 then in the membrane of the synaptic vesicle, and this is going to pump 5-hydroxytryptamine, but also other monoamines such as dopamine and noradrenaline, which we'll come back to later when we do the noradrenergic neurons. Okay, so VMAT2 can pump all the monoamines, not just 5-hydroxytryptamine. It's just that we're talking about a serotonergic neuron, so we're talking about serotonin. Now VMAT2, so I'll put VMAT2 here since we're talking about a neuron. It is not a primary active transporter. It is instead a secondary active transporter. Okay, now what does that mean? That means that it is not going to use ATP to do this job. Okay, it's not going to use energy from ATP to move the serotonin into the synaptic vesicle lumen. Instead, it is going to use some sort of ionic gradient. So an important thing to understand is that there is a very high concentration of protons in synaptic vesicles. Okay, and that's because of a transporter for protons on the surface of the synaptic vesicle membrane. Okay, so coloured in green, supposedly there is a proton pump which pumps protons from the cytoplasm into the synaptic vesicle lumen, and this will use ATP when it does that. Okay, so it will be hydrolyzing ATP to ADP in inorganic phosphate. Okay, so on the surface of the synaptic vesicle you have these proton pumps which are pumping protons into the lumen of the synaptic vesicle, okay, using ATP. Now, that is a form of primary active transport, so the proton pump is a primary active transporter. Okay, but now the vesicular monoamine transporter 2 doesn't need to be a primary active transporter. What it can do is it can allow protons to leave the synaptic vesicle lumen, going down their electrochemical gradient, and in exchange for those protons which it set free, which will release energy as they move out of the synaptic vesicle, it's going to use that energy to move the serotonin into the lumen of the synaptic vesicle. So in fact, VMAT2 will allow 2 protons to leave the synaptic vesicle lumen for every serotonin molecule that it moves in. Okay, so that's how you fill synaptic vesicles with serotonin molecules. Okay, right. So we'll call it there for this video, and in the next video we'll continue our discussion of the serotonergic neurons by discussing the release of serotonin from the serotonergic neurons and then the reuptake of it.